Good morning and good afternoon. My name is Brent Halper, and I'm the Scientific Director for the AI Horizons Network, and this is our weekly seminar series. This week we have Kristen Severson from the Health Analytics Research Group in IBM Research Cambridge. Kristen is a postdoc in that group, and she received her PhD from MIT and also has a bachelor's in chemical engineering from Carnegie Mellon. Kristen will be talking today about unsupervised learning with contrastive latent variable models. Sorry about that, Kristen. And ask people to hold their questions for the end if possible, since you're coming in muted. If it's necessary to ask, please unmute yourself or type your question in the chat. Without further ado, Kristen, it's all yours.
Great. Thanks so much for the introduction, and thanks everyone for joining. As Brent mentioned, today I'll be presenting our work on unsupervised learning with contrastive latent variable models. And this is a collaboration with my colleagues Soumya Ghosh and Kenny Ng, both also in the Health Analytics Research Group at IBM Research Cambridge. This work was presented at AAAI 2019. And so to get started, I first want to just clarify what I mean by unsupervised learning. So the technique, contrastive latent variable models, that I'll present today is meant to function in a space where we don't have a clear objective. So there's not a particular prediction task or classification task, but instead we're exploring a data set hoping to find interesting patterns or structures. Oh, sorry, oh, there we go. Okay, so for those of you who have worked in an unsupervised setting before, you likely know that one of the first things you might be interested in doing is visualizing your data set. Particularly when you have a high dimensional data set, it's not trivial to simply look at every dimension. So a very popular technique is principal component analysis, or PCA. And what PCA does is it maximizes the retained variance in some lower dimensional space. Ideally, that lower dimensional representation will then reveal some structure that you can relate back to your application. So here on the right-hand plot, I'm showing an example. These are samples that came from a single-cell RNA-seq data set. And we've projected the data using PCA into two-dimensional space. And looking at it, perhaps there's some amount of interesting structure as we kind of move maybe to the upper left. But it's difficult to say unless you have some additional information. Alternatively, we can use the contrastive latent variable model approach and get this two-dimensional representation of the same data set. And again, perhaps now kind of in the lower middle region, we're starting to see some structure, but it's not obvious at this point. So in doing this, one thing we might hope to have is some additional orthogonal information. So here now I've colored each sample based on whether or not it was pre or post-treatment. So these are single-cell RNA-seq samples from patients who have leukemia and have underwent stem cell therapy. So once we've added this information about pre and post-treatment, we see that the projection of the contrastive latent variable model approach does learn this structure that separates those two groups, whereas the PCA projection has those two groups directly layered on top of each other. And so we have a hypothesis as to why this might happen, and that's that high variance directions aren't necessarily relevant to whatever domain application we're working in. So we might be interested in characterizing some expected baseline variation so that we can search for variance directions that differentiate our data set of interest. In effect, we're looking for patterns that are enriched in one data set as compared to a second data set. And I have a figure here to kind of demonstrate this in a simple two-dimensional case. So here the green dots represent our single-cell RNA-seq leukemia patients. There are these two classes, but a priori we don't know about that. So we see one class here and the other class here. If we take that data and project it using PCA, the direction we'll learn is shown by the solid black line. And when we project our points onto that line, we see that the two classes collapse on top of each other. Instead, if we have some kind of background or baseline data set, in this case, we considered healthy control patients, single-cell RNA-seq data, we can characterize some amount of expected variation and kind of project our data set of interest into the remaining space. So the direction we'll learn in that case is instead this dotted line, and we project our green patients or leukemia patients onto that. We maintain this subgroup structure. So we didn't get something for free here. In order to use the contrastive latent variable model approach, we needed to have a second data set. And however, I think that there are many natural settings where these types of pairs of data sets occur. So in the leukemia example, we had control or healthy populations versus a study population. We might be interested in pre versus post intervention groups, or perhaps we have signal or signal-free measurements where we have some characterization of the instrumentation we're using to gather our data set. So despite the somewhat natural setting of looking for patterns that are enriched in a data set as compared to another, there aren't a lot of algorithms that can work with sets of data, specifically where we don't have any pairwise relationship between samples in those sets. So a notable example, a notable exception to this is the recently proposed contrastive PCA technique, which was proposed by Abid and his collaborators. And they propose this method that works in exactly this setting where we have some data set of interest, which we'll call the target data set, whose samples are indicated by X sub I, and some second background data set whose samples will indicate Y sub J. And what CPCA does is it forms an empirical covariance matrix. That's the empirical covariance of the target data set subtracted and then minus the empirical covariance of the background data set with some parameter to tune the difference alpha. Once we've formed this kind of type of empirical covariance, then we can use the singular value decomposition algorithm to learn the lower dimensional space as we would in a classic PCA algorithm. So this is a really interesting technique, but it is deterministic. And so we were interested in instead learning a probabilistic approach. And we motivated that for several reasons, some of which I've highlighted here. So probabilistic models have the advantages of being able to incorporate prior information. They have natural handling for noisy and missing data. They can perform model and feature selection. And they also can be incorporated into larger probabilistic systems. So given this background, our goal was to learn a model that would achieve similar aims as the contrastive PCA approach, but done so in a probabilistic framework. So again, we're hoping to search for patterns that are enriched in a target data set as compared to some background data set. And here in the blue box, I'm showing the structure of the model. So our Xi's are going to be samples from our target data set, and Yj's are samples from our background. I'll point out that here there's no pairwise relationship between I and J, and there's no requirement that the number of samples must be equal. So N doesn't necessarily equal M. What does need to be the same is the number of the type and number of measurements. And what we're going to do is we're going to project this data into two spaces. For the target data set, we have a shared space and a private latent space, whereas in the background case, we only have this shared space. So the factor loading or transformation from the lower dimensional space to the full dimensional space is shared amongst these two groups, whereas the target latent space is private to only our target data set. So there is no T sub J for the background samples. And then the rest of the parameters are data set specific means and noise terms. Here, the dimensionality of Zi and Zj is the same, and we have the ability to choose the dimensionality of Ti. In both cases, those values will be less than D, and the dimensionality K plus T also will be less than D. Okay, so before I go into details on the contrastive latent variable model approach, I thought it would be a good idea to just provide some amount of background on latent variable models more generally. And so here, what I'm interested in is some probabilistic model that's a joint distribution over our observed variables X and some unobserved or latent variable Z. So we can consider, how we would write that distribution, where we're going to marginalize over our unobserved variables. And kind of the questions that we're interested in answering are, what can we say about the probability of the unobserved variable given the observed variable? And how might we make predictions about some new sample X star given what we've already seen? If you're familiar with this kind of plate notation, this model is shown here on the right-hand side. Shaded nodes are meant to indicate observations that are observed, whereas unshaded ones are unobserved. And this plate with an N means that's the number of samples. And the directed edge means that Zn is a parent of Xn. So to give a particular example, you might be familiar with the probabilistic extension of PCA, which is just known as PPCA. And that model looks like this here, where we have our observed data X sub n, which we're going to project into some lower dimensional space Z sub n using the transformation W. And it has the corresponding distributional assumptions. Our latent variable is assumed to come from a standard normal distribution. And then we have a conditional distribution of the observed data, such that the mean is specified by WZN plus mu, and the noise is some sigma squared I. Then using the properties of multivariate normal distributions, we can also consider the marginal distribution of the observed data X. So there are a lot of variations of these types of models. I've just highlighted a few here. You can think of different characteristics, so they might be discrete, continuous. You could have deep models where you have nonlinear relationships, or you might use linear ones. They can be parametric, where you've specified the particular distributional assumptions, or they could also be non-parametric. So some examples that you might have heard of include mixture models, factor analysis, hidden Markov models, latent Dirichlet allocation, Gaussian process latent variable models. But this is just a small set of this really rich space of probabilistic models. So in order to use these, we need to learn the distributional parameters. And for a small class of models, the properties are such that we can learn them using expectation maximization. And what expectation maximization does is it maximizes a lower bound of the log likelihood. And in order to do that, it requires calculating the posterior estimates of the latent variables as well as the derivatives of the likelihood with respect to the parameters. For many types of latent variable models, this isn't tractable, and instead variational inference is used. What variational inference effectively is an approximate posterior over this P of Z given X. So in variational inference, what we do is we say we're going to assume that posterior comes from some variational family of distributions that will approximate using Q. And then we're going to fit the parameters of that distribution phi to be close to the exact posterior. So graphically, we have this space of variational approximations. We initialize our parameters at some point, and we use some optimization routine to get optimal values where we're as close as possible to the true posterior, where that distance is measured by the KL divergence. So we can't do that directly, because if we could measure the distance to the true posterior, that would mean we had access to the true posterior. So instead, what we'll do is we'll optimize the evidence lower bound, or ELBO, sometimes also called the negative variational energy. And that's a lower bound on the log of the probability of X. So I haven't shown it here, but optimizing this P of x is equivalent to minimizing the KL divergence that I showed in the previous slide. So we end up with this objective function, and we'll use coordinate ascent to alternate between optimizing the parameters of our variational approximation phi and the true parameters, or the parameters of our model theta. There are several different types of variational inference. I'm not going to go through all of them today, but just to give some examples, we can consider a stochastic variational inference, which uses subsets of the data to get noisy estimates of the gradient. This can be important because the parameters of our variational inference scale with the data set size. We might also use black box variational inference, which is uses an expectation of the gradient of the elbow, then samples from it. BBVI is nice because it doesn't require us to derive model-specific quantities, despite writing down, outside of writing down the model generally. So BBVI is the method we've employed for the contrastive latent variable model. It gives us the most flexibility in choosing our distributional assumptions, and that is implemented in Edward, which is a Python package built on top of TensorFlow. The kind of key insight of black box variational inference is to write the gradients as expectations. There are two main techniques to do that, reparameterization gradients and score function gradients. In our case, we're using reparameterization gradients, and that's because our latent variables are continuous. Just one more comment on variational inference before I go back to the model. So I mentioned that our parameters were growing with the data set size, which can lead to intractability. So another type of approach might be to perform amortized inference. So here we had our initial family of distributions that we're using to approximate the true posterior. We might instead consider a subclass where now instead of having a phi for each I, we have some mapping that takes in our observation xi as the parameters. So now we just need to learn this one global f of theta as opposed to learning parameters for each distribution, and this can help as the number of samples in your data set grows very large. Okay, so coming back to the contrastive latent variable model, we can consider the relationships here graphically, where again, xi are samples from our target data set, and that's the data set that we're interested in learning about, and yj are the background data. Again, yj only has one latent space and has representations z sub j, and our target data has two latent representations, zi and ti. Primarily, we're going to be interested in visualizing what these ti's are. Once we have that model, we can write down the full probability distribution. So here we have our parameters theta. We have our conditional likelihoods and our marginal likelihoods, marginal distributions of our latent variables. So the primary modeling decisions are to select the likelihoods as well as the priors on the loading matrices W and S. Again, W is the loading matrices that's private to the target space, and S is shared across the target and background. Again, many choices of these priors will lead to distributions that aren't tractable, which is what motivated our choice to use black box variational inference in solving for the parameters. So before I go into all the kind of different variations you can have in this model, I want to demonstrate this using a simple one. So contrastive latent variable models look very similar to PPCA when you use Gaussian variables to model the latent representations and a Gaussian likelihood. So again, the major difference is the idea that now we have these two spaces. So if we go back to the two-dimensional example I showed before, we can see that when we project our green target points of interest onto the PCA direction, we get the representation shown here on the right. So we're in a two-dimensional space, so we can really only project into a one-dimensional space. And we see that that representation is completely overlapping. Here, now I've colored the two clusters as red and blue. The idea being that we might have some orthogonal information that would allow us to do that coloring, but again, this is just projecting only the green points. If we instead use the contrastive latent variable model approach, we see that we recover these two distinct groups in our 1D space. Okay, so now what I'd like to do is go through a series of examples that demonstrate some of the ways you can alter this kind of base model to get different functionality. I mentioned, or I motivated this work in part using the contrastive PCA work that was done, and all of our examples here were taken from their paper and then showing kind of how you could do the analysis slightly differently if you have these probabilistic formulations. So this set of examples is motivated by the CPCA work. So the first example I'll show is, again, this same problem using single-cell RNA-seq data. Here, just some insight into kind of what that data set looks like. There are approximately 8,000 target samples and approximately 2,000 background samples. So again, I'd like to stress that there's no pairwise relationship between the patients or participants in these studies. And the observed dimensionality is 500. So for those of you familiar with single-cell RNA-seq data, that already has some amount of preprocessing performed, such that our starting dimensionality is only 500. And so I alluded briefly to the fact that in these models, we still need to choose the dimensionality of the latent representation. So for anybody who's used PCA, you know you need to pick how many principal components you want to use in learning this lower dimensional representation. And so one thing we can do with the probabilistic formulation is to use an automatic relevance detection prior. This is particularly convenient as our dimensionality grows, like this, 500. You know, we need to choose the dimensionality of the shared space, and you know, the kind of classic way to do that would be to sweep over all dimensionality, which can be time-consuming. It can also be very difficult to choose kind of at what point do we eventually want to truncate. So what we've done here is this ARD prior has been shown to cause some columns of the factor loading matrix to go to 0, which in effect automatically chooses the dimensionality. That prior is specifically here over our shared factor loading matrix, where for each column of the matrix, we have a variance specified by alpha j, and that has a prior that's inverse gamma. We initialize S to be d-1 columns because we know we at least want to reduce the dimensionality by 1. And in this particular example, I've fixed the dimensionality of the target space to be 2, and that choice is motivated by interest in visualizing the target space. So we don't need to make that choice, but here we've decided a priori that dimensionality is 2. And here we're using a Gaussian likelihood and this variational approximation. And what I'm showing in the right-hand plot is the cumulative percent variance explained by the columns of the shared factor loading matrix. So what we can see is we reach a sharp cutoff in the case where we have the ARD prior, where the dimensionality of the shared space is approximately 100. Now, if we don't use ARD and we have a dimensionality of d-1, we see kind of this smoothly increasing percent variance explained, which then we have to, choose how much or how little we want to truncate that. And then leveraging the factor loading matrix that was learned with the ARD prior, we get the representation shown here on the left, which does in fact recover these pre and post treatment groups. The left-hand side is only visualizing the target data set, these T sub I representations. So nowhere here am I showing what the kind of background space looks like. Okay. So another functionality we can get out of a probabilistic formulation is robustness and missing data handling. This example here is subgroup discovery using mouse protein expression data. So here we had a target sample of 270 mice samples and a background sample of 135 mice. The background samples are known healthy control mice, and the target is an unknown mixture of healthy and trisomic mice. So trisomic mice are used as mouse models for Down syndrome. And our data set is 77 protein measurements. So here we add a inverse gamma prior to our noise term. And that's useful because the resulting likelihood is now a student's t distribution. And as compared to the Gaussian distribution, student's t has heavier tails. And what that will do will allow our model to be slightly less influenced by outliers in our data set. So if for whatever reason we think we might have outliers, we can use this to add robustness against those samples. And then I'm showing the results here. 1.6% missing data probably seems like a strange choice. That is the amount of missingness that's occurring in this data set naturally. So I mentioned these are publicly available data sets, so that the authors cite technical artifacts there for the reason some data is missing. And we can handle that missingness by marginalizing over those missing observations. So the probabilistic formulation allows us to do that naturally. And we see that in the target representation, we do, in fact, recover these two distinct subtypes. Then I looked at artificially increasing the amount of missingness, and we can see that even up to 75% missing data, we still, for the most part, do recover these two groups. Obviously, they're becoming noisier as we have more missing data, but I find this technique to be nice because it doesn't require you to a priori choose some imputation technique such that you're able to use kind of like a classic PCA or the CPCA algorithm. Another functionality we get by using this formulation is feature selection. So I'll demonstrate this using a heterogeneous sensor data set. Here we have about 6,500 target samples and about 3,000 background samples. The target data set here are participants who are either doing a cycling or squatting activity while wearing a bunch of sensors. And the background samples are participants wearing those same sensors but just lying still. And there are 23 different sensor measurements, which are of 4 different types. There's an accelerometer, an ECG, gyroscope, I think, and magnometer. And so we were interested in asking the question, can we learn the subgroup representation potentially without using all 4 of these sensor types? And the way we looked at doing that was to penalize the rows of the target factor loading matrix such that our latent representation would be a function of only a subset of the measurements. So here I'm showing a penalty inspired by a group lasso type of penalty, which will cause full rows of our factor loading matrix to be 0. And so when we apply this penalty, what we find is that all of the magnometer contributions go to 0. Here I'm showing the row index versus the row norm. So you can see for the green magnometer points, those are all 0. But using this factor loading matrix and looking at the projection, we're still able to learn these 2 distinct groups of cycling and squatting. So this type of analysis helps us to learn that if we wanted to differentiate these tasks, again, we might not bother to use the magnometer measurements. I'll point out that I did this using just a regularization type framework. We could also consider how we would do this using distributional assumptions. Things like the horseshoe prior can also achieve these types of results. Okay, so one last extension of this is a nonlinear extension. So the structure of our model really makes it simple to extend the analysis to a nonlinear setting. So here we again have our 2 groups of data, the target and background data sets. But now instead of having those linear mappings, S and W, we've now added nonlinear mappings, which are parameterized by neural networks. The model has the same structure where there's this shared mapping and then a private target mapping. And so well, let me first give just a diagram of kind of what this model structure looks like then. So we have our encoders, then we're projecting into this lower dimensional space where, again, the target has this additional flexibility, and then a shared decoder and a private decoder for the target only. And then our reconstructions of the target space will be these additive contributions from both spaces. So here we again chose an example from the CPCA paper to demonstrate this. Here we have noisy image data where our target samples are 5,000 samples of MNIST digits, which have been overlaid with ImageNet graphs. And the background samples are just ImageNet graph samples. The observed dimensionality here is 784. So I have some examples here. The top images would be samples from the target data set, and the background are just these graph images. And we can investigate the target latent representation as we're showing here. And we see now we've colored each point by the digits 0 to 9, and we see that there's nice consistency in this latent space amongst these digits. As I should have been saying throughout, I didn't really bother to show the PCA embedding of these different results, but they either can't find them at all, or in this case are much less consistent in terms of representing the digits. And I think really one of the nice properties of this particular model is because we had those two decoders, we can also consider generative samples. So here, these are samples from running just the target decoder, and these are samples on the bottom here of running just the shared decoder. So these are, digits that we're able to generate, and then these they have some amount of patchy resemblance to kind of what we might expect graphs to look like. So I think this kind of disentangling behavior is a really nice feature of having this model structure. So to conclude, we've proposed this idea of contrastive latent variable models that are really meant to help you discover patterns that are enriched in a data set as compared to another. It has a lot of applications, including data exploration, visualization, as well as pre-processing if you're trying to reduce dimensionality before some other modeling effort. We think doing this problem probabilistically and as opposed to deterministically gives you a lot of nice properties. Here today I've shown robustness, sparsity, as well as nonlinear variation. We have some ideas for future extensions. We might consider larger numbers of data sets, because for now we've just considered these pairs, as well as data types. We haven't really focused too much on kind of how to best handle counts or categorical information. So I mentioned that this work was presented at AAAI 2019. I checked this morning, and I don't think the proceedings are out yet, but we have uploaded the full paper and supplemental information to archive. So you're, of course, welcome to check it out there. And I'm happy to take any questions.
Thank you very much, Kristen. Now's the time. If you have questions, please unmute yourself. It's the little red microphone at the bottom of your screen.
Thank you, Kristen. It was a great presentation. I have a question. Maybe you mentioned in the presentation earlier. So what is the difference between CPCA and the one you propose? I believe yours is probabilistic model and the other is not.
Yes, that's the main difference. And I'm not trying to make a claim here that I've proven that they're equivalent, just to clarify, but they do achieve similar aims, with the major difference being that CPCA is deterministic and leverages an SVD algorithm, whereas this is probabilistic and leverages black box variational inference.
Under which application? So is there any, under some settings, your method will perform better as opposed to theirs?
Yeah, so I think better is always a tricky question when you're talking about an unsupervised learning problem. I think because of the probabilistic formulation, you have some additional capabilities. Like for instance, the feature selection, it's not there might be ways to do feature selection in the CPCA deterministic framework. It's not kind of as readily obvious to me, but that doesn't mean it doesn't exist. So it's more about the different properties of the model than necessarily saying one is better. If you look at like applying CPCA to the single cell RNA-seq data, you will get the same, or you can get the same representation depending on the dimensionalities of the spaces you choose.
Okay, got it. Thank you so much.
Yeah.
Okay, any other questions? Going once? Going twice? Okay, Kristen, thank you very, very much for your presentation. You had almost 24 people on in real time. I'll remind the audience that the tapes of these, the recordings of these presentations are now available on YouTube at ibm.biz/AIHN-recordings. And next week, our speaker is Hong Wang from UC Santa Barbara, speaking about sentence embedding alignment for lifelong relation extraction. That'll be on Tuesday, May 28th at 2 p.m. Eastern Time. So, Kristen, again, thank you very, very much, and thank you everybody who has attended.
Thanks for having me.